Okay, so first of all, I would like to thank the organizers for giving me an opportunity to talk at STATPHYS meeting. So I'm going to talk about how matrix viscoelasticity controls the spatio-temporal tissue organization. And this work has been done in collaboration with Alberto and David. They did the experiments, and the model has been developed by me and Damon. So here you can look at two images. Uh, so both of these images are of tissue. They are composed of the same cell type, but the only difference is that when they are growing, they have been grown in a different environment. So the extracellular matrix in one case is more elastic, in other case viscoelastic, and that only that difference leads to a very different kind of tissue growth. So that's what I'm going to talk about, how changing the environment around the tissue can lead to different kind of growth. So why it is important to focus on mechanical properties of the tissue. So of course, tissue rigidity determines the proliferation of the cells, uh, cell differentiation, motility, development, tumorigenesis and wound healing. And of course, there are many other things which we do not know about and we'll learn as science progresses. So this is an example of tissue growth in the case when we have uh, a different kind of environment around the tissue. And here, from left to right, we are changing the stiffness of the extracellular matrix. And you can see just by changing uh, the stiffness of the matrix, it leads to different kind of tissue growth. So in all these four images, in the beginning, the tissue is made out of the same cells, but if you wait for some time, and if the matrix environment is different, you can see the growth looks completely different. So till now, most of the time, the focus has been only on tissue rigidity. No one has focused on how the fluidity or viscosity of the material is going to affect. And in past 10 years, past 10 to 12 years, a group of David Mooney and others, they have been focusing on how to develop the materials, artificial materials, which can mimic the actual tissue type, such as one can bring viscoelastic, one can mimic viscoelastic extracellular matrix, and how independently one can control the viscosity and elasticity of these materials. So here we have, this is the stress relaxation plot. So this is a PAM, which is very commonly used in developing a 3D tissue, which is highly elastic, or you can say the time scale to relax is much longer. This is collagen, which is used to develop mainly 2D to do 2D cell culture, which is highly fluidic in nature. Time scale is to relax is much smaller. And these are the tissues in our body, such as adipose, liver, and brain. And we can see that relaxation time for these tissues are in between, which represents that it is viscoelastic in nature. So, so the focus is only to see how viscoelasticity affects the this tissue growth. So these are experiments done in the lab of David and Alberto. So what they have done, they have considered an elastic matrix and a viscoelastic matrix. And when I say elastic and elastic, the only parameter which I'm changing is the viscosity. So in case of elastic material, the viscosity is very high. In case of viscoelastic material, viscosity is very less. It is more fluidic in nature. So you take the stem cell type, you make a tissue out of it, and then you wait and see how the tissue growth looks like. So you can see that in case of elastic uh, extracellular matrix, the growth is very smooth, spherical, it's a controlled growth, and it is always maintaining a stable interface, a smooth interface. Whereas if you look at the tissue growth for the viscoelastic material, in this case, the growth is much, there is an abrupt growth. You can also see that there is finger formation starts to happen, and the growth is much more uh, uh, faster and abrupt compared to the elastic case. One can also measure, quantify it. So they have looked at they have taken the 2D section of these tissue, they have looked at the area, and they see that in case of viscoelastic matrix, the growth is much stronger. Also, one can quantify the circularity, and they see that for elastic case, it is able to maintain its circular structure, whereas for the viscoelastic case, it is losing its circularity. So to model this, we developed this mathematical model where we are considering a passive matrix, which is viscoelastic in nature, and a viscous, active viscous tissue, which is proliferating. So to model the passive viscoelastic material, we are again considering a uniform a monodispersed bed, and the dynamics is given by this overdamped Langevin equation. We control the viscosity through this parameter mu m. This potential is consist of Lennard-Jones elastic, simple harmonic elastic kind of model, and then there is cell substrate interaction, which is purely repulsive in nature. Then we have for the tissue, again, we have overdamped Langevin equation. We have, since those are these cells are active in nature, we have a kind of random motility here. The interaction potential is consist of Lennard-Jones and cell substrate repulsion. We control the motility through this parameter m, you can see here. And now about proliferation. So for proliferation, we have two rules. A cell is going to divide only if it is older than a certain time tau g. And once a cell division takes place, then you look at the energetic criteria. You see whether the stress is increasing and decreasing. If the stress is, or the total energy of the system is increasing, you accept it with certain probability. So this is how the model looks like. Now, in this model, what are the control parameters? We have tissue viscosity, we have proliferation rate, we have motility. For the matrix, we have this matrix viscosity, which we are controlling. Most of the time, we keep the elastic coefficient to be fixed. And this proliferation is local stress dependent. So basically, what happens? We have motility. When cells are motile, they will displace the extracellular matrix. They will create the free space. Once there is free space, then there is possibility for proliferation. When proliferation starts to happen, and if it is faster than the time scale of the matrix, then there is a different kind of morphologies come into picture. So if you take this model and simulate it, and in one case you keep the extracellular, the viscosity of extracellular matrix to be very large, and in other case the viscosity to be very small, then this is how the growth going to look like. So this is the growth of the tissue in a extracellular matrix, which with very high viscosity, which is elastic, which we call elastic, and in the other case when it is viscoelastic. So you can see here we have also quantified how the area is going to look like. For the elastic case, the growth is much weaker, whereas for the viscoelastic case, the growth is much stronger. Again, we quantified the circularity, and you can see that for the elastic case, it has smooth interface, whereas for the viscoelastic case, it is losing its circularity. Now, as I said, there are other parameters such as motility and proliferation. So we try to control these parameters independently. So now in this case, what we did, we reduced the motility, and then we tried to see how it is going to affect the overall growth. So this is the control case where we are not changing any of the parameters. This is the case where we are killing the motility for both the elastic and viscoelastic matrix. And we can see that in this case, the growth looks very similar in both the cases. The growth is, of course, suppressed, but also interface remains smooth. In experiments as well, using, I don't remember the name of the drug, using a drug, you can control the motility. And they also observed the same thing. This is the case when you kill the motility. This is how the overall area going to look like. These are the corresponding images. These are some statistical significant. I don't know. Means you can measure the. Okay, maybe I can describe it later. So this is from the experimental data. So again, you can control the. In the same way, you can kill the proliferation. You keep the same level of motility, and when you kill the proliferation, you again observe that you keep the same level of motility, all the properties to be same, and just by changing the removing the proliferation, you see that again, since there is no growth, only motility is not able to disturb the this interfacial structure. And again, using this drug thymidine, they can kill the proliferation, and they observe the same similar thing. Okay. Till now we have talked about the viscosity, motility, and proliferation. We have not talked about stiffness. So as I was mentioning in my second slide, that stiffness also controls the proliferation. In this paper, they have mentioned it clearly that as you increase the stiffness of the matrix, that is also going to affect the proliferation in a positive way. So what we are saying that the means in a positive way, and also they have looked at the motility of the cell, how it is going to affect the motility. So usually there is a sweet spot when you increase your stiffness, your cell motility also increases. So what we are doing now, we are saying that our cell motility is going to be a function of the stiffness of the matrix. And if you do that, this is how the dynamics is going to look like. So this is for a soft tissue where its stiffness is smallest. This is for the intermediate stiffness, and this is the, yes, thank you. This is for the strongest stiffness. And you can see that as you are increasing the stiffness, the growth is becoming stronger and stronger, and also circularity decreases. Here we have quantified volume. Since we are doing simulation, we can easily do both volume and area. So here you can see for the viscoelastic case, the growth is fastest. Viscoelastic stiffest extracellular matrix. This is the intermediate stiffness. This is for the weakest stiffness. And this is the case for the elastic matrix. Again, you can look at the sphericity, and the same is also getting reflected in the circularity. So this is again in the experiment, they have found something similar. Now I will go to the final. Okay, you can do the same exercise with motility and proliferation. If you kill the motility, again, it is not going to, it is independent of what is the matrix around it. Same for the proliferation and motility. So what one can do a phase diagram based on the viscosity ratio, cell flux, and cell activity. So cell activity is directly a measure of motility, how motile your cells are. Cell flux is an emergent quantity. How the proliferation, what is the proliferation rate for a given system. So if you do that, so what we realize when, so this is the interface between a stable interface and unstable interface. Inside this green boundary, we have a stable interface. Outside this boundary, we have unstable interface. So you can see that when your proliferation rate is very small, you have a stable interface. When your viscosity ratio is very small, which means that a viscosity ratio is defined as tissue viscosity to matrix viscosity. When your matrix viscosity is very large, which means matrix viscosity is very small, you have a stable interface. Again, the cell flux is directly related to cell proliferation. So when your cell proliferation is very small, again, it is stable. And outside this, it is unstable. So with this, I will conclude. And these are just conclusions. Thank you. So now just one more statement. So now till now we have considered uniform matrix, but in future we are thinking of making it, we are changing the property dynamically, and also we are bringing in active degradation. Thank you.